Our group at MIT works on technologies for imaging and controlling biological systems, and increasingly we're focusing on trying to see how we could help with the problem of aging and longevity. We've done some work collaboratively on the aging brain, but what I hope to convey today is that a bunch of these tool sets are maturing, and if we could integrate them, maybe we could build extremely detailed models in the computer to help us understand where in aging one might intervene. That's really what motivated me to enter the field of neuroscience. I trained as an engineer and was obsessed with this idea that you could understand what thoughts and feelings were, but it's a big mess, right? And so my sort of overarching goal was let's build technologies to see and control the building blocks of the brain, and then we could apply them to build models of how brains work. But of course, all biological systems are made of the same kinds of things. And so up till now, we've shared the tools very widely, but increasingly we'd like to integrate them into a pipeline. Now, why are these things so hard? Well, there's obviously a couple issues. One is the spatial complexity of biology, from atom to molecule to cell to tissue to whole organism. Then you have the temporal complexity, from millisecond all the way to lifespan. And then thirdly, and maybe most dauntingly, you have the heterogeneity of biology, right? Unlike physics or chemistry, where you have to deal with a small number of objects and a small number of ways they interact, in biology, the number of building blocks, genes, gene products, and biomolecules is a deliriously long laundry list. And so today I'll talk very briefly, just at an overview level, of a couple technologies we built that try to confront spatial scales, temporal scales, and sort of molecular heterogeneity at those fundamental levels head on. So let's start with spatial scales. You know, wouldn't it be great if you could map a whole organism even down to nearly atomic scale? That was the motivating question that prompted us to try to build a nanoimaging tool that we call expansion microscopy. Now, why do we build this? Well, there's lots of methods for nanoimaging, and nanoimaging is very helpful because the building blocks of life, molecules, are nanoscale objects. But all nanoimaging methods are difficult to apply to large 3D objects like the brain or a whole organism. And so in our group, we often think about trying to do the opposite of what people are doing. And so starting with two very talented grad students at the time, Fei Chen and Paul Tilburg, we thought, let's take biological specimens, infuse them with basically baby diaper polymer, swellable materials, very densely and very evenly. And if we do it just right and add water, the swellable polymer will grow as it absorbs the water, and it'll make the biological specimen larger. So we call this expansion microscopy. And there's not time in these short talks to go through all the details of the chemistry, but suffice it to say that you can apply chemicals in a certain series of steps where you form a densely permeating hyperswellable hydrogel throughout a specimen. You anchor biomolecules to the gel, you soften it up, and then when you add water, you can pull those biomolecules apart. So in panel B here is a piece of the mouse brain, several millimeters on the side, so a pretty macroscopic object from a nanoscale imaging standpoint. Panel C, about a day later or so, we've blown it up 100 times in volume. And at this point, we can blow things up 1 million times in volume or more. And so the polymer starts out very dense, like the cartoon in the upper left. You know, the polymer spacing is about 1 to 2 nanometers, so smaller than many biomolecules. And then you could extend it to a large conformation, like in the lower left. So conceptually, what's happening at the nanoscale? Well, we're taking a cell like this brain cell on the left and making it look like something like this thing on the right. So it's a constellation of biomolecules hovering in space. If two biomolecules were touching, now there's some minimum distance apart. And if you have two biomolecules that are some distance apart, it's scaled up by a linear factor. That's by design. We also spent a lot of time in the early days validating. So we do pre-expansion imaging with a classical nanoimaging method, and then post-expansion imaging with a conventional microscope. Expansion process is not perfectly even. You get a few % distortion over a typical microscope field of view, but it turns out that for the vast majority of biological and medical questions, that's totally fine. We are working to make the expansion process more even, by the way. But even with the original expansion factor, which is 4.5-fold, and the original few % distortion, there are lots of things that we could do that were difficult and sometimes even impossible to do with earlier tools. So here on the left, we're zooming in from top to bottom. Each white square is zoomed in the panel below. I guess I can use the mouse pointer. And the right hand is the same as the left in terms of specimen and microscope, but after we expand it. The color code is shown on the left-hand side because in light microscopy, we're decorating key biomolecules with fluorescent probes. And I hope you can see that the right-hand column is much more clear and crisp than the left-hand column. So we can now make nanoscale measurements on ordinary microscopes. That's lesson number one. We can democratize nanoimaging and make it so that you can scan fairly large volumes with nanoscale precision with ordinary hardware that's universal in biology already. Okay, that's looking at natural proteins. You can also do labeling of different things artificially. So you can take fluorescent proteins engineered from corals and jellyfish, and you can express them in a barcode-like fashion. So you can label cells or even molecules in interesting combinatorial fashion. So here we are just expressing fluorescent proteins in different cells because we wanted to make a map of the brain. And if you zoom in to this little white square at the top, it's fuzzy, right? Because the wiring of the brain is very, very tiny. But the upper right, same brain specimen, same microscope, but after we expanded, you can now map the fine wiring of the brain. And so many, many groups now are using expansion plus some kind of color code or stain in order to map brain circuitry. That's all protein imaging. When Oswasi joined our group, and later Gaoqi Yang and Yi Kui made a very inexpensive version of this, we started looking at nucleic acids. So we can build anchors that bind to different kinds of biomolecule, DNA, RNA, proteins, lipids, and sugars. And then you form the polymer that binds those anchors, add water and the polymer swells, and through those anchors or handles you pull everything apart. And so here you can see individual messenger RNAs shown in pink. So now we look at proteins and nucleic acids. So starting to map out lots of different components is possible, right? The genome and its shape, RNA, proteins, and so forth. You know, it's a multiplexable way of mapping out lots of things at the same time. Now, how big can you expand? So far, I've showed you expansions about fourfold, but you can take a specimen and expand it, form a second polymer in the space opened up by the first expansion, and expand it again. So you can get exponential spatial amplification with a linear number of steps. It's sort of like PCR, but for space. And so this is work done by J.B. Chang, Dablina Sarkar, Jinyan Kang, and Oswasi. We can take a specimen and expand it over and over again. Now, this offers a second superpower, which is shown in this cartoon. So when, again, in light microscopy, you're imaging fluorescent labels that you attach to things, right? So this red and green molecule, red and green dyes are staining proteins on the outside of a complex, but they can't get inside, right? If you pull them apart, though, now the yellow and blue labels can get inside and turn those previously invisible molecules into visible ones. So now you can start to map out nanoarchitecture of very complex protein assemblies. Of course, in aging, we look at lots of protein aggregates. Here we are looking at amyloid plaques with Li Wei Sai at MIT. Of course, they're very densely packed. We wonder, are we missing something by staining them when they are jam-packed together? So the yellow images are where we stain with antibodies against A-beta-42 pre-expansion. These are mouse models of Alzheimer's, but we see similar things in human tissues. And then the purple, same antibody, same specimen, is when we relabel after we expand. We've decrowded all the proteins away from each other, and we can label them better. So this, of course, works in other things than just brain. Yongshen Zhao and others in our group showed that we could expand and label all sorts of stuff, prostate, lung, breast, pancreas. On the left are normal, and on the right are cancer-containing specimens obtained from human biopsies. So the take-home message here is that by expanding biological specimens, we hope to be able to map all the building blocks of life. We would like to make maps down to nearly atomic scale. One of our collaborators, Silvio Ruzzoli, in Germany, has shown that the expansion method is precise down to sub-nanometer scale. So you can maybe even see parts of a molecule. And he has a paper coming out soon which talks about how to do that. But suffice it to say that this is a very easy-to-use technology. It's spreading very rapidly throughout biology. I think the number of experimental papers and preprints is about to hit 800 in a day now, so it's really simple to do. And so if we can really make a map of what's happening in aging down to nearly atomic scale, our hope is to really be able to look at how molecules interact and to be able to maybe make new hypotheses for what could be causally involved with aging. Of course, then you have to go back and do old-fashioned causal perturbations to test that. But my hope is that this can be a hypothesis generation strategy. Now, I think, as must be all too obvious, one of the obvious limitations of expansion is that it doesn't work on living things, right? This only works on preserved, meaning dead, specimens. That's because you're pulling all the building blocks of life apart from each other, right? Molecules work by touching each other. That's how they bind, cut, modify, and so forth each other. We've pulled them all apart. So the living state has been halted. And so in the last part of the talk, I would like to tell you about the imaging of living things. I'm going to skip over optogenetics. That's kind of old stuff and mostly published long ago. But I want to talk about the imaging of things when they are alive, and then I'll end by trying to stitch it all together. If you could image a lot of stuff when a cell is, or tissue is alive, and then map out structure, could you build computational models, which is what we'd like to do going forward. All right. Part of the problem with understanding things when they're alive is that there's a lot of stuff going on. So this is a cartoon stolen from Wikipedia. Here's the cell membrane shown as the orange boundary on the left. Here's the nuclear membrane in blue. I think we've all seen diagrams like this, right? Cells take in countless things, hormones and nutrients and all sorts of stuff. Inside the cell is this roiling ocean of computation where kinases phosphorylate and transcription factors move and the cytoskeleton rearranges. And then eventually the cell generates some kind of output. Maybe it lives or dies, or maybe it moves or stays put. And each of these arrows, of course, is probably an entire PhD thesis. And a lot of these parts are worked out in different systems, right? So maybe one part of the system's been worked out in a fruit fly, and another part's been figured out in a mouse. But in any given cell, how do we know what of this is actually going on? Now, the good news is many people have brilliantly created fluorescent reporters of different signals, right? So you could take a fluorescent protein from coral or jellyfish, you can connect it to, let's say, a calcium binding protein, or a kinase binding protein, or a cyclic AMP binding protein, et cetera, and people have ingeniously made many fluorescent binders of different kinds of biomolecular signal. This is a screenshot from Jin Zhang's wonderful database from her group at UC San Diego. But what if you want to look at lots of things at the same time? Well, in the past, you would take a blue sensor or a red sensor, and you could get a couple colors, right? And you'd build a couple color microscope, and you'd have blue would be indicating calcium, and red would indicate protein kinase A or something. But what if you want to look at a lot of stuff, right? So we set forth this challenge. And again, we really believe in the ease of use of technology. So we wanted to build a technology where people could image an arbitrary number of things in a living system. But again, without needing exotic hardware, anybody should be able to do it. And ease of use matters using fully genetically encoded reagents. And so, one of the ideas that our group often takes a classical engineering idea and we implement it through chemistry, right? So, expansion microscopy, we're doing nanomechanical engineering through chemistry. And so we thought a lot about it for many years and realized, hey, cellular telephony, telecommunications, the internet, there are lots of examples out there where there's lots of channel sharing, right? You have lots of communication going on in the same space. Could we implement that through chemistry? And so we took two ideas that are basically how the internet and cellular telephony work, spatially multiplexed strategies and temporally multiplexed strategies, and we implemented them through chemistry. Okay, so what's spatial multiplexing? Well, in this building, there's lots of Wi-Fi antennas. As you walk from room to room, your cell phone or your laptop is being picked up by different antennas. And so this building can support much more Wi-Fi bandwidth than if you just had one antenna that we all had to fight over, right? So what if we had the same thing in a cell? What if different places in a cell could tell you different stories while it was alive? And then temporally multiplex imaging. So our cell phones, even if we're all in the same place, all are sort of using similar frequencies, but they have different temporal codes. And that's how we can all share the space in this room, and we could all probably make calls right now if we wanted, and they wouldn't interfere too much with each other. So how can we implement these things through chemistry? Okay, spatially multiplex imaging. So Shannon Johnson and Cheng-Yang Lingu worked out a way of doing this. What if you could take fluorescent reporters, like the kind I told you about earlier, and park them at different places in a living cell, random places, but stable places? So in this cartoon, the points labeled 1 could report, let's say, calcium, and the points labeled 2 could report, let's say, protein kinase A. So while the cell's alive, each little dot will blink out a movie of what is happening there, and you could imagine lots of dots all over the cell telling you different stories of what's happening in the cell. And then when you're done with your experiment, you preserve it, and then using techniques like I talked about in the first part of the talk, where you can stain an image and so forth, you can figure out which reporter was at each point. So here's how we make it work. We can take different fluorescent reporters and fuse them to different self-assembling peptides. So these are proteins that are kind of like the coat proteins of viruses. They self-assemble into clusters, except they're artificial, so they're not going to mess up the biology. And so here is the take-home message here. We took 3 different off-the-shelf reporters, one for calcium, one for cyclic AMP, and one for protein kinase A. And these are very interesting molecules involved with many kinds of signals, not just in aging, but in learning and memory and all sorts of stuff. And we fused them to different self-assembling proteins. So while the cell's alive, you get these green images. They're all green reporters, by the way, so this works even if everything is the same color. And then the false color images are what you get when you're done with your experiment and you stain to figure out which sensor is at each point. So the take-home message is we can image many different signals by having these reporters at slightly different places in the cell. Here we are looking at a whole bunch of three different signals downstream of giving forskolin, a drug that makes cyclic AMP. And I don't want to get into the details here, but I did a PhD on learning and memory, so I really wanted to see if we could understand how a short-term stimulus to the brain might result in a long-term change, like a memory. These are cells in a dish. This is not memory, but we were able to relate the speed of one signal, calcium, to the amplitude of a second signal, protein kinase A. And so there's lots of, I think, intellectual analogs to this kind of question in the field of aging, where you want to know where some short-term insult might have resulted in a long-term change. Now we can map out how those stimuli might relate to each other. Now, why do you need this? Well, if you image calcium in one set of cells over here and protein kinase A in a second set of cells over here, you would miss out on the relationship, right? But here you can just read out the relationship by looking at it. We can add a fourth indicator, a protein kinase C indicator, or a fifth indicator, an ERK kinase indicator. Anyway, so we were kind of getting into the technical weeds here, but the take-home message is we can take different fluorescent reporters of signals and park them at different points in space, and cells are big compared to molecules. So the hope is that we can eavesdrop on a very large number of signals and map out how a cell takes its inputs and transforms them into outputs. All right. Well, what if you don't have an off-the-shelf reporter ready to go, though? What if you just want to know whether a gene is on or off? Or what if you just want to know where a protein is? In the last few minutes, I'm going to try to explain a technology that we published fairly recently, and to be honest, I'm still figuring out how best to explain it. But maybe the best analogy would be a piece of music, right? So a flute has a high pitch, and a tuba has a low pitch, and then the violin or whatever has like a medium pitch. And the way our ears work is that different pitches end up at different physical locations in our ear, right? The way our cochlea works inside our head is that high-pitched sounds go to a different place in our ear than low-pitched sounds. It's doing a transform and sorting out the frequencies. So the core idea here is, what if different cells, signals in a cell could have different musical instruments? You could take a brief movie of the cell and do the same transform that our ear does and invert it and get all the signals. So we can't make musical instruments that small, but it turns out that there are fluorophores that flicker back and forth when you illuminate them in the right way. And so in work that Yong Chen did when he was a postdoc in our group, we worked on reversibly switchable fluorescent proteins. So what are those? Well, if you shine blue light, one of them might glow green and go dim. Shine purple light and it'll come back again. So you can toggle it on and off, right? A little bit like a musical tone fluctuating, right? So these come in many different flavors with different temporal properties. Here we have 6 different greenish fluorophores that have different flicker time constants. If you just shine constant light, then they don't flicker back and forth, but you get a decay. And these decays are linearly independent, the same way that musical instrument pure tones are linearly independent. And for the aficionados, this is not photo bleaching or fluorescence lifetime. This is flickering. But this is a reversible and safe process. Okay, so I tried to explain it by the analogy to music. I might try a little bit of a more technical explanation now, just to try out a second version of the story. Here are a couple cells in a dish. What if each point in the cell has a couple different fluorophores that flicker at different frequencies? Maybe they indicate different amounts of a gene. Now, if you shine constant light, they'll show a characteristic decay. And if you take a brief movie of the cell, then every pixel of the cell will be a linear combination of those exponential decays. And the weight in the sum is just the amount of signal there, which you can get with a linear inversion. So I tried to explain it two different ways, one by analogy to music, one through getting into the math a bit more. I hope one of these at least resonated with you, but maybe just showing you some data would be better than trying to find a third way of explaining it. Okay, so we got 6 different greenish fluorophores. They decay at different rates. We take a brief movie and do an inversion. Okay, here we took 6 different proteins that go to different parts of a cell. Lysosomes, endoplasmic reticulum, nucleus, mitochondria. Each one has a greenish protein with a different flicker time constant. Take a brief movie, do a linear inversion, and we can get all 6 parts of the cell. And it only took one color, right? This is a green microscope. So you can imagine having an almost unlimited number of things tagged. Take a movie and invert it, and you can see those things. Here's the cell cycle. I'm sure most people in this room know more about than I do. We just make the tools here. But there are proteins that turn on in different parts of the cell cycle. We can fuse them to different green proteins with different flicker time constants, take brief movies and at different points of the cell cycle and invert them, and we can map the cell cycle with a single color. So the point I'm trying to make with this last part of the talk, which is out of time, is that we can take different engineering ideas, such as those that are used in wireless telecommunications and in the internet and so forth, make molecular versions of them, and our goal is to image many, many, many things at the same time in a single living cell, so we can know how cells convert their inputs into their outputs. And these are all genetically encoded tools. They don't require any hardware that are beyond the scope of a traditional biology lab. Now, where do we want to go next? Up till now, our group, over the last 18 years of our existence, have shared the tools very freely. Expansion microscopy is in use by, as I said, probably thousands of groups by this point. Our optogenetic tools, which I didn't have time to talk about, are also very widespread. But now we want to start integrating these things. Suppose that we can image lots of cells in a living cell or tissue or even organism. We will image lots of signals in those cells over time. We can then preserve the specimen, do expansion, and map the molecular architecture of those cells, tissues, or organisms. Can we combine these into mechanistic models and infer what kinds of processes are occurring? And so we're working with a large number of collaborative groups now to try to build the machine learning architectures to do those assemblies, and we're very excited about that as one of our main focuses going forward. So we share all of our tools freely. Please visit our website, synthneuro.org. And as we engage in this new adventure and become more and more into the computational realm, we're looking for more and more collaborators who would like to work on specific problems with us, or who would like to try out a different AI or machine learning approach. So please stay in touch.
Thank you very much. Got time for 2 or 3 questions.
That's pretty fantastic. I was curious about extension microscopy for the chromatin. Those pieces of DNA are connected to each other. And so you expand on them, you'd expect some of them might break. Mm-hmm. I'm sure you'll look at this kind of in some details. Can you comment on that, please?
Yeah, great question. So I think to expand the genome and preserve its architecture, one should do a controlled fragmentation, ideally. In ChIP-seq or Hi-C or lots of other techniques, you do a controlled fragmentation, right? Often with a restriction enzyme. You can imagine taking the genome, anchoring it to the polymer, chop into pieces, and pull the pieces apart. And I think that if you were expanding very big, that would be ideally the way to do it. It's not my group's expertise, but I hear rumblings of this possibility. A group at Yale did a small expansion, like 4-fold expansion. And for small expansions, you don't have to do this chopping because any perturbations would still be too small to see. So they had a beautiful paper still showing that even with a 4-fold expansion, they call it chromatin expansion microscopy, you can derive some very interesting scientific insights. But I think as we expand to very, very large volumes, yeah, I think a controlled fragmentation will probably be required.
Hi, here, under the TV. Hello?
Yes.
Yeah. So if you combine 2 fluorescent tags in the same protein, can you expand the range of flickering detection that you can do? And in that sense, could you, label all proteins inside the cell almost?
For the second part of the talk?
Yeah. Yeah, just because you show that you have one flickering signature for one fluorescent protein, what if you have 2 fluorescent proteins that are different in the same protein sequence?
Yeah. Yeah, so this idea of using fluorophores as computational devices is an intriguing idea. What if you could have multiple fluorophores? You can imagine sending energy to one, there's some coupling between them, and if each of them has their own dynamics, you could imagine a chain of these, yeah. I think there's a lot of possibilities. Yeah, there's just too many things to do and not enough time. So, but yeah, if you're interested, we should talk more about that.
One more question.
Sorry, to sort of build on that, can you measure events like phosphorylations or dimerizations or this sort of thing?
We can measure phosphorylation with the first of these technologies. So here we're using a protein kinase A indicator, I think from Xin Zhang's group, and then we can park these different indicators at different places, and then we can measure phosphorylation at those different places. For the second way of doing it, actually the second way can be used as well. So Yang had this great idea to use so-called KTRs, kinase translocation reporters. They move around when they're phosphorylated. And as you might guess from the way that this technology works, it's very good at seeing when proteins move around because every point is being decomposed into its sum. So I guess both techniques could be used for that.
Fantastic. Thank you very much, Ed.
Great. Thank you.